Thank you, Marita, for inviting me to speak to you this morning. So I'm going to be speaking to you about how our research group has exploited the genomic information and how we've applied that to better understand sports injuries and the molecular mechanisms underpinning these sports injuries, well, some of these sports injuries. So connective tissues, the injuries that we've concentrated on have involved musculoskeletal connective tissues, and these are tissues which are found around your muscles, around your cells, around your muscles and in bones, ligaments and tendons, as well as bones and cartilage. The two common, there's several musculoskeletal soft tissue injuries which are commonly affected, and they commonly affect structures. These common structures are commonly affected during injury, which as a result from participating in occupational activities within the workplace or physical activities, such as your sporting activities, and they can broadly be classified into two categories: your acute onset injuries, and examples of these are your injuries of your knee, your anterior cruciate ligament ruptures, your overuse injuries. An example that we've studied is your overuse Achilles tendinopathies. Your acute injuries usually result from a specific macrotraumatic event, while your overuse injuries result over a period of time, resulting from cumulative microscopic damage to the tissue, and this repetitive damage results in microtraumatic events, and the volume of injury and damage to the tissue results in symptoms. Looking at the structure of both tendon and ligaments, they are very similar, and they are mainly collagenous structures and are composed primarily of water and tightly packed parallel bundles consisting predominantly of type 1 collagen. These fibrils aggregate into larger structural units, and they finally produce your tendon and ligaments. So we've been very fascinated at what are the mechanisms causing the structural damage within tendons and ligaments, and how that therefore affects in symptoms. So there are receptors on our cells, and they are unique in that they have the ability to respond to force and load. So here I've shown you these receptors on the cells, and they send a stimulus then back into the cytoplasm to upregulate several cytokines such as your interleukins, and they are able to set off a whole pattern of a cascade of events which activates other molecules and which are involved in tendon remodeling, allowing your tendon and ligament to respond to the load, allowing for adaptation to the load and force, and ultimately repair. And so if any one of these processes is not working correctly, one can understand therefore why an injury would result. But the million-dollar question is, what are the most important biological markers in all these different pathways leading to injury? So one of the questions we've also asked is the cell unable to detect load or force, or is it unable to respond to that load or force, or is it able to respond to all of that but is unable to repair it? So if there's an inappropriate response to the force and load, the cell is unable to repair and correct the building blocks such as your collagens, your glycoproteins, or your proteoglycans. It is also unable to, these are all hypotheses, unable to control the regulation of several enzymes such as your matrix metalloproteinases, and these are critical in degrading the damaged tissues such as your collagens, and there are other markers which are responsible for down-regulating these enzymes so that you can get enough synthesis of your collagen molecules. And all of these mechanisms could potentially be involved or be altered, and therefore some people are at an increased predisposition to specific soft tissue injuries. So if you look at the tendon pathology as a continuum model, so all of us respond to load or force, but we also have this individual variation in how we appropriately respond to this load and force and how we adapt. And if we all have different thresholds of responding to load and force. And the one model suggests that if you are unable to react normally or respond proactively to a force or a load, it will eventually, you won't be able to repair your collagens, you have an increase in proteoglycans, and there's also an increase of vascular growth, and then at the end result will be a degenerative tendon and therefore tendinopathy. However, you can have an intervention at some point where you prevent the progression to degenerative tendinopathy, and where your cell is able to react appropriately, it's able to synthesize all the appropriate molecules to repair the damage, and you have a healing as well as an adaptation response to your force. The slide shows us there's also continuum in how people, and variation of how people respond to force and load. You have an extreme case where some people, where you have a maximum tolerable load, reaching that you have an increased risk of sustaining an injury. Where here in the middle you have a no injury, so you have normal force that you can respond to, and the other extreme is when you also have no force is also not a good thing, so at that rate you also have an increased risk of injuries. So musculoskeletal soft tissue injuries are multifactorial in nature. There's not just one risk factor, but there are many risk factors, and it's actually the interaction of these complex risk factors which result in the clinical presentation or the injury. And we are only looking at one component in our research, and that is genetics. We're exploiting genetics as a tool to understand what are the potential molecular mechanisms which are going wrong. Some of the risk factors which have been identified or suggested are intrinsic, that's within the body, and others are extrinsic, which is outside of the body. And it's the combination of your intrinsic risk factors which predisposes an athlete, and together with your external environment makes the athlete more susceptible, and only once an inciting event occurs will the injury result. So there's no single cause resulting in the phenotype. There have been several studies on families which have suggested that there is a genetic component and there's an increased risk within families for these soft tissue injuries, such as in the case of rotator cuff tears as well as ACL ligament tears. Just to recap, I know you've had an introductory lesson. What's the difference between a mutation and a polymorphism? A mutation is often used to identify a disease-associated sequence variant, and if we can equate that to, whereas a polymorphism is usually associated with it's common in the population and it's not causative of the disease, but it's associated with the disease. And here's an example. If this sentence can be equated to a gene, and this L over here is the position of the mutation within that gene, that insertion over there, or the alteration in that position, results in an altered gene. For example, pulp changes that L being replaced with the M, changes that word into pump, which is a completely different word to pulp, and thereby we can use this analogy that a mutation alters the gene so that it makes a new protein or it affects the protein. So the function or the interpretation of that gene is completely altered. Where in the case with the polymorphism, here at the bottom, if you replace that one letter in that word or within that gene, it doesn't change the meaning of the word. So similarly, a polymorphism in the gene does not change the meaning of the gene, but it could potentially affect how much of that protein encoded by the gene is made. So that's quite important to understand. And polymorphisms may contribute to not to severe diseases, but may influence characteristics such as your hair, your height, hair color, eye color, and also susceptibility to disease. Where mutation, if you've got a mutation, you're most likely to get a certain particular disease, such as your inherited conditions. Example is your Ehlers-Danlos syndrome. This picture is here to illustrate to you that although the human genome sequencing has shown us that we are 99.9% similar, if we look at all the individuals on this picture and around you, we are very different. And it is believed that it's this 0.1% difference in our genetic makeup which is responsible for us, for our biological variation, and for all of us to be looking differently. And so this highlights to us that we are individuals and that there's a lot of biological variation in how our bodies function. So we've exploited the human genome and we've used several polymorphisms across the genes, and we've identified polymorphisms to be associated with particular soft tissue injuries such as chronic Achilles tendinopathy and ruptures of the anterior cruciate ligament, both within South African and Australian populations. So now I'm going to focus on one particular gene as an example of the several loci that we've identified. And this gene is the COL5A1 gene, and it encodes for type 5 collagen. Type 5 collagen is one of the essential building blocks of the collagen fibril, and it is essential for life. Loss of both copies of your type 5 collagen gene results is embryonically lethal, and it is found that type 5 collagen regulates fibril formation as well as diameter, so it plays a very important role in the rate limiting step of fibril formation. And there's an inverse relationship with type 5 collagen content within a tissue and the fibril diameter, such that if you have a higher type 5 collagen present, your fibril diameters will be smaller. Mutations within this gene has also been associated with the with Ehlers-Danlos syndrome, which is a connective tissue disease, and you require both copies of your COL5A1 gene to be functional. This cartoon over here illustrates how type 5 collagen intercalates with type 1 collagen to form your collagen fibril bundles. And there are many other proteins also involved in regulating this fibrillogenesis, which is the formation of your fibers of your tendons and ligaments. So this initial association we found with a particular polymorphism at the 3' end, which is the last bit of the COL5A1 gene to be associated with Achilles tendinopathy. And subsequent analysis, we've looked at 3 or 4 other polymorphisms within the 3' end of this gene, and we found a similar pattern where these genotypes have been associated with an increased risk of Achilles tendinopathy, while the CC genotype at this particular locus has been associated with reduced risk of Achilles tendinopathy. So our research is showing us that there is something at the 3' end within the COL5A1 gene, that there's some sequence motif in that region which is associated with an increased risk of these injuries. What it is, we're still trying to find that. So the question is this region functional? Because why are we finding all these associations? Finding these associations does not directly implicate that type 5 collagen is directly involved with the biological mechanisms of this injury. We need to explore this further with functional analysis so that we can identify the biological mechanisms, and that's what we have subsequently done. Our group has cloned the 3' end of this into expression vectors, and using luciferase activity, they found that those individuals who do not have the injury have a specific sequence region or sequence motif within the 3' end, which is very different to those who are affected. And we found that if you have this particular sequence within the 3' UTR of the COL5A1 gene, your messenger RNA is more stable, and we hypothesized that with a stable messenger RNA you have more type 5 collagen produced from these individuals with a risk DNA motif. Interestingly, bioinformatics tools have also shown that if you have the control genotype motif, you have a completely different structure of your COL5A1 3' UTR end compared to your tendinopathy genotype profile. So the data is hinting that there seems to be a functional relationship with the 3' end of the COL5A1 gene and risk for Achilles tendinopathy. Interestingly, there's other research that have also now been shown that polymorphisms within this region is also associated with injuries within superficial digital flexor tendinopathy within race horses. They've also linked these polymorphisms with joint laxity and mechanical properties of tendons. So the evidence is becoming more and more available, supporting that there seems to be a functional activity within that region of the gene. We've also recently, it's unpublished data, it's still in review, looked at the polymorphisms within this particular region, and we found similar associations with an occupational injury such as carpal tunnel syndrome. So the proposed mechanism is that if you have a particular, for example, this is your control genotype profile, DNA profile at the COL5A1 3' UTR region, and this is your risk profile, which was found in the tendinopathy patients. We found that if you have this risk profile, you have a more stable messenger RNA, and we hypothesize that this leads to more type 5 collagen produced. The more type 5 collagen produced, the tighter your fiber bundles and the more densely packed they are, compared to the less, in the contrast, where you have the opposite DNA profile. You have less type 5 collagen being produced. Your collagen fiber is less packed and the diameters are much wider. So the difference in the structural properties of the collagen fiber may lead to differences in biomechanical properties of the tendon fibers between these different individuals. So this one region within the genome may play a role in contributing to ligament and tendon injuries. Remember that it's a multifactorial condition and it won't just be this particular locus. So we've looked at that one region, but we've also looked at several other loci or regions within the genome, and we found independent associations with polymorphisms within the collagen 1A1 gene, which encodes for a component of type 1 collagen. And these are as I said in the beginning, that they form play a very important structural role in tendons and ligaments. We've also found polymorphisms within the microRNA binding domain of MER608. And particularly with Achilles tendinopathy. MIR608 interestingly binds to a region within the COL5A1 gene. We've also found polymorphisms associated with Achilles tendinopathy within the GDF5 gene, which encodes for a component functioning the cell signaling pathway. And we've also found 2 polymorphisms within the caspase-8 gene, which is involved with programmed cell death and apoptosis. So all these, we hypothesize that each and every one of these proteins play a role in fibrillogenesis and so if we look at the individual and all the different polymorphisms, we think there's a genetic continuum, is that you have at the extreme end, you have severe mutations which are rare in the general population, and they can result in either a lethal phenotype, as I mentioned, the COL5A1 mutations. If you knock both copies out, it's lethal. If you have one mutation, it can cause Ehlers-Danlos syndrome. You can have other variations where they have minimal effect, or you can have on the other extreme, in the other extreme event, where you have common polymorphisms with a greater environmental exposure can result into injuries such as your ligament or tendon injuries. So how can individual variation lead to soft tissue injuries? Well, taking into account the individual's medical or clinical history, the injury profile, their physical activity, one can look at the genotype, and together with all of this, we can stratify individuals into high risk categories, intermediate risk categories, or low risk categories. Once you know the individual's risk status, you can therefore design or manage that individual's training or rehabilitation programs much more better. However, the sports injury gene screen is not diagnostic. It's also not predictive. It consists of 8 DNA sequence variants within 5 clinically useful genes, and they collectively, with all the other risk indicators, can be used to identify or stratify the individual into certain risk categories. We'd like to highlight that there is no single cause for these tendon and ligament injuries, that it's a multifactorial condition, and it's the interaction between all the risk factors which results in the injury. So the genetic test is not performed on its own. It needs to be interpreted together with the individual's clinical history, its physical activity history, as well as any other risk indicators. If you look at the sports injury gene screen test, these are the 5 loci that are tested and the 8 polymorphisms that are tested. The cells in red indicate the high risk scoring genotypes. The cells in green are your protective genotypes, and the cells in orange have no effects to date. So if there is risk scores come back, as an example, a risk score of for Achilles tendinopathy, your risk score can go from 0 to 8, and for anterior cruciate ligament ruptures, your risk score can go from 0 to 2. So if an individual has a risk score of 2, you can say that they have a reduced risk, and if an individual has a 0 risk, it has an unaltered risk based on its genotype profile alone. However, I'd like to stress that you cannot base your total risk on only the genetics, but it has to be interpreted together with the other clinical indicators. So together with your risk score, you will also get a table summarizing all the other non-modifiable risk factors together with the modifiable risk factors to alert the clinician what to look at for. For example, if the person is at an increased risk, then these are the ones that you would be able to modify, such as your physical activity program can be adjusted. Your factors such as body mass index are easily modifiable as well. So this DNA test is not diagnostic in nature, and there are important medical ethical issues to be considered, that you cannot guarantee that if an individual has a certain genetic risk score that they are going to develop a high risk score. That doesn't mean that they will develop that particular injury, or doesn't mean that they have a low genetic risk score that they will not ever develop that injury. It also doesn't predict when you're going to sustain a ligament or tendon injury, and problems will arise if individuals are selected for or against using this genetic risk score. These are some of the case reports that we've had in the past. That individual, a clinician has contacted our research group and said he sustained an acute pectoralis major insertion rupture and recently has sustained a acute complete Achilles tendon rupture. He's not on any quinolone antibiotics, nor does he have any previous history of this. And there's also a negative family history. Can we perhaps guide him as to the possible risk factors and also how to reduce his risk in the future? Does he think he carries the genetic markers? Okay. So this was the response. It's an interesting case. Our understanding of acute tendon injuries is not as well defined as for chronic injuries, because all the risk factors that I've illustrated to you before from our genetic research is based on chronic Achilles tendinopathy. But the principles are probably the same. It is reasonable to say that the player is predisposed for acute tendon injuries, being the second acute injury that the patient has suffered from. We cannot pinpoint the exact predisposing factor, as this injury is also multifactorial in nature and is more than likely to result from a combination of several genetic and non-genetic risk factors. However, we can say that the individual is at an increased risk, taking the clinical history into account of acute musculoskeletal soft tissue injuries, and thus the medical team should be alerted that he is more than likely also predisposed to other acute musculoskeletal soft tissue injuries, which commonly occur when playing X amount of sport, and I can't label the sport in this case. We're not the experts in this particular clinical phenotype, but the obvious injuries to consider which may be avoided through preventative training may include X and X, and therefore it is the best strategy at this point in time. And the respective prehabilitation exercises required to reduce these injuries of concern are once again up to your expertise. Research has however been published for most of these injuries proposing interventions which have been scientifically shown to reduce risk. So the patient clearly is at an increased risk of musculoskeletal injuries, and therefore management has to be careful at his training program and also probably suggest prehabilitation training program to prevent subsequent injuries of this nature. Another case, if an individual came, he's 52, he's male, he's overweight, and he lives a sedentary life and wants to start running socially to get fit and lose weight, would he be at an increased risk of sustaining a musculoskeletal soft tissue injuries? For example, if we did his genetic profile and he comes up with a risk score of 6, we know that he would be at an increased risk of 4 and greater would put him at an increased risk. And taking into account his age is also an intrinsic risk factor for an increased risk of these types of injuries. Overweight is also a risk factor, so he has a strong clinical risk factor together with his genetics risk factor, puts him at a very high risk for sustaining this type of injury. So therefore, his management of what type of exercise he does needs to be controlled. His weight needs to be controlled, and because of his increased risk. But his genetic background doesn't guarantee that he will be predisposed to a musculoskeletal soft tissue injury, but take into account his other clinical parameters, he's most likely to, if it's not managed appropriately. So I hope I've shown you how we've harnessed the genetic information to try and understand biologically how these different mechanisms cause the ligament and tendon injuries, and how we've applied that into a genetic test, and how the test can be interpreted cautiously to better manage these individuals. Thank you.
Any questions? I think you've explained it so very well and comprehensively. So there's one question.
I'm still confused about the practical application. Any of our national sports teams where this activity utilized? Is it ever about the sportsmen? Is that a genetic in the risk?
That's an excellent question, and we often hear that. The genetic test is not there to say you are at a high risk and therefore you should not be playing these sports. But it's to show you that genetically you're already at a high risk, and take into account all your other risk factors which we can modify. Let's reduce your chances of getting an injury by giving you prehabilitation exercises so that we monitor you more carefully and prevent possibly you rupturing your ACL or your Achilles tendon. Are any of our national sports team using this device? Not yet, but we have had international appeal for the test already. And there's loads. I haven't presented this in my talk, but for the Olympics, the previous Olympics, the British team had their athletes all tested. Yeah, so it is happening all around the world.
Microphone then, please, just for the sake of the recording.
I just want to answer, Doctor, on that specific question. My name is Lou Jane, and I've spoken to some of our Springbok rugby players, and I've spoken to some of our Protea cricket players, specifically regarding to this test. And most of the ones that I've spoken to have indicated that they would love to do this test specifically to be preventative towards their training in future.
Yeah.
So they've definitely indicated to me that they would be very interested in doing something like this. They're not even scared that they won't be chosen because of this. For them, it's much more important to be preventative in future for injuries, so that they can actually stay in the teams much longer than they would have been if they didn't know the outcome of such a test.
And there are several publications in previous years of soccer teams in the UK who have gone for testing as well.
Just to add to that, a question: in the case of very, very highly paid professional players, could this be a potential source of discrimination, or do you think it could actually be an issue? You said in our players it may not be the case, but I think if you're talking about these multi-million sort of people, could that be a problem?
Could potentially be a problem, but I think clinicians realize that these are multifactorial conditions and that your genetic makeup is just one component, but if you know your risks, then you can reduce your risks.
Thank you, Alison. I think what we need to start understanding is that just like we test the cholesterol level or hypertension, a genetic risk factor is just adding on to that. So it is very permanent, so one have to be very careful to decide whether you need to do the test or not. But if one see it as part of a bigger picture and not thinking that the genetics will now override all the other things would be the only answer, we know that life doesn't work like that. Medical applications is very integrative, and doctors look at many things. And I think it's not so difficult to understand that if you just add it to all the other risk factors, it could provide information, but one should not misunderstand it. And I think that is where we that we say we want to work as teams, scientists, medical doctors, dietitians, even bioinformaticists that we have on the teams, so that everybody could bring their knowledge to the in the end produce a report that can go back to that doctor. So we are also very careful not to offer these types of tests directly to the public. So that is why it would be a slow thing, but it is the way that it should be done, because there needs always be someone that can explain even the report that we as scientists try to write to the best of our knowledge. There's always so many things that of that specific patient sitting with that doctor that you can't capture in a report, and that doctor, and it's also who gets referred for these tests. It's a whole process. It starts there, that even the people that get selected for genetic tests must be so carefully selected so that the information wouldn't do more harm than good. And I think what Alison has explained, it's all about what is environmental, what can we modify, because your genes you cannot modify. And we know that there might be many other genes that could override single effects that we test for, but like many people also say, we have to start with what we had and build up until the day that we would have for 1,000 rand maybe your whole genome sequenced, and then be able to align that information with a clinical picture going forward.